Hello, my name's Jade, and I am from a small town in Nebraska. Normally, I don't tell people that because it's a bit of a conversation killer. People are usually like, oh, yeah, I've driven through there once. But I promise I'm telling you this now because it's relevant to this talk. So if you are one of those people who have driven through Nebraska, this is probably what you saw, just fields and fields of corn. And to do this level of intensive monocropping agriculture requires that we add things to the soil, namely nitrogen. But there's also a downside to adding nitrogen to the soil, and that is that sometimes nitrates will leak into the water supply, which is what happened in my hometown, leading to the EPA to actually sue my hometown. And growing up, I thought it was perfectly normal to not be able to drink water from the sink because it's poisonous. But it's not normal and it's not right. And there are ways that we can overcome this challenge. So one of those ways is to transition from using synthetic fertilizers, which are based on fossil fuels, to working with sustainable fertilizers, which are based on microorganisms. So why haven't we done that yet? Well, there's certainly several companies who are working on it, but in general, when we add back these microbial inoculants, they don't always get a reliable result. And that's part of the reason is because we don't quite understand the molecular mechanisms that are underlying those plant microbe interactions. And that's exactly what we're trying to do in this project. So in this project, which is led by Sharon Doty out of the University of Washington, we're studying poplar, which is a bioenergy crop. You might know it as a cottonwood tree. And Sharon's group actually goes out to these poplar stands where the poplar is doing very well despite being in a less than ideal condition. So maybe it's in a droughted area or a nutrient deficient soil. And she isolates microbes from these plants to look at how could these microbes be helping the poplar survive and thrive in this less than ideal ecosystem. So one of those microbes that Hello, thank you. Okay. Sorry about that. So one of the microbes I'm talking about today is called Burkholderia vietnamiensis, or the strain name is WPB, which is how I'll refer to it today. So this is a aerobic bacteria that can live inside the plant or outside. It's also a nitrogen fixer, so it has this N-fixation gene, which is encodes for a nitrogenase enzyme. It's a pretty conserved gene. And what Sharon's group did was they added this GFP promoter to the N-fixation gene so that whenever the bacteria are expressing the N-fixation gene, they will light up green. Interestingly, what we saw was that the bacteria don't always light up green. Sometimes even in the same colony, some cells are expressing N-fixation and others are not. And so we don't really understand, we wanted to understand why that was happening. The first thing we did was quantify how many cells are actively transcribing N-fixation at once. And so we worked with Will Chrysler at EMSL to do fluorescence activated cell sorting. And Will found out that only about 11.5% of the WPB cells are actively expressing the N-fixation gene. So we wanted to understand why only some of them are expressing this gene. So next we tried fly-fish, which is a spatial transcriptomics method, and Da-Hong He at EMSL helped us with this technique. So in these images, the white blurry dots are the bacterial cells, the blue dots are fluorescence, but the red circles are actual N-fixation transcripts. And so what we really were kind of looking here at was whether or not the presence of nitrogen in the media was going to influence how many N-fixation transcripts the bacteria produced. But what Da-Hong found was that actually it mattered more whether the cells were aggregated together versus just individual planktonic cells. So when the WPB cells were aggregated together, they produced about 30 times more N-fixation transcripts. So to us, that kind of hinted at maybe there's some sort of quorum sensing going on, that's cell-to-cell signaling, telling the microbes to turn on this gene. So just because they are turning on the N-fixation gene doesn't necessarily mean that they are producing this nitrogenase enzyme, and actually fixing nitrogen. So we wanted to confirm that. We used nanosims. Thank you, Jeremy, for the introduction to nanosims. We worked with John Cliff at EMSL, and he got this really beautiful image showing these individual WPB cells that have a different amount of 15N enrichment. So these microbes are fixing 15N2 and incorporating it into the cells, but there's a bimodal distribution in how much they're actually fixing. So this method sort of confirmed that the fixation of nitrogen is variable within this bacterial population. So now we wanted to understand, okay, so once the microbes fix the nitrogen, what are they producing that they could be giving to the plant? And so we worked with Youngmo Kim and Natalie Munoz at EMSL to do 15N metabolomics on these samples. So we grew the microbes for a 9-day time course on this 15N2. And then Natalie and Youngmo found 16 compounds that were enriched significantly over time. And 15N, a lot of those compounds were amino acids, which is not super surprising because the bacteria really needs those to survive. Some of them, though, were some of these polyamines like ornithine, davarine, putrescine. And those are compounds that the microbes could be using for N storage that they could then be giving to the plant. So that was an interesting finding. Also, a part of this study, we worked with Amy Zimmerman to do 15N proteomics. And so Amy looked at which peptides were enriched in 15N as well and cross-referenced those to the metabolic pathways for this microbe. And that confirmed that the microbes are sending that freshly fixed nitrogen into those amino acid biosynthesis and polyamine biosynthesis pathways. So the microbes are really doubling down on that nitrogen fixing metabolism. Interestingly, Amy also found that the microbes are directing this 15N to peptides in the quorum sensing pathways. And so that kind of adds evidence to what we're thinking about how quorum sensing has this influence on nitrogen fixation for these bacteria specifically. All right. Okay. Okay, so next we looked at actually going in and understanding why there's this spatial difference where some cells are expressing NFH and others are not. And so we actually cut out these areas that were green, expressing NFH, and sent those through metabolomics. And then we also cut out the areas from the same colony that were not fluorescent and sent those through metabolomics as well. So it turns out that the cells that are actively actively expressing NFH are those that are more and have more of this polyamine group, so cadaverine and putrescine. And then the areas that are not actively expressing NFH are actually producing more carbon compounds like trehalose sugar and citric acid. So what we think is going on here is sort of a division of labor where the cells that are not actively expressing NFH are providing some sort of carbon source to those that are, because nitrogen fixation is very metabolically expensive. Okay, so everything up to now is just the bacteria by itself, but we also wanted to see whether or not any of these things were happening when we added the bacteria to the plant. And so to do this, we worked, Daisy Herrera and I worked on culturing the poplar plants in the rhizochip platform, which is one of EMSL's capabilities. And we inoculated these plants with the WPB and looked at them under a microscope. What we saw was that the WPB, these little green dots, were still actively expressing the NFH gene within the root cells, which is these blue boundary layers there. So that's something that we hadn't known for sure whether or not that was going to happen inside the plant. So that was an exciting result for us. And we used 2 different techniques to confirm that the bacterial cells were actually inside the plant. So one was lattice light sheet imaging with de Hong, and that gave us these 3-dimensional images. And I don't think my video is going to play. But you can see the cells, the bacterial cells are actually within the plant cells. Similarly, we used confocal to scan down into the root to see that they were inside the boundaries of the root. Oh, it does play. There we go. And this confocal also told us that the cells are really only in the outer, about 30 microns of the plant root. So we think they're only in the epidermal layer. One thing that surprised us was that we found these strange features in the root cells, these little spheres, that we don't see when we don't add the bacteria. And inside the spheres is these fluorescent NFH expressing bacteria. And so what we think is happening is this could be some physical mechanism that the plant, maybe it's like corralling the bacteria inside the roots, and by doing so is helping to enhance the expression of that NFH gene. So we're calling these nitrosomes. We're doing some follow-on studies now, but sort of an exciting result too. And so now we know a little bit more about what the bacteria could be providing for the plant, but now we also want to look at what the plant could be using as signaling molecules to the bacteria. So we were working with Kevin Zemaitis and Dusan Velichkovich to do multi-mass spec imaging of the rhizochip. And we have seen that some metabolites tend to stick closer to the roots where others diffuse further away from the plant. And Kevin and Dusan are able to get really good coverage of the poplar's secondary exometabolome with this. So right now we're working on correlating which metabolites are in the same area as the bacteria to try to understand which molecules the plant could be using to signal to the microbes. Finally, I want to talk about another project that we're doing some spatial imaging of plant roots on. For this project, Vimal and Tanya Winkler grew these poplar plants with and without endophyte inoculation and under drought or well-watered conditions. And then Robert Stanley and Dusan Velichkovich used MALDI to map the metabolites on root cross sections. And so far, this is very preliminary data, but so far what we're seeing is that some metabolites do seem to only be present when the plant has been inoculated with the bacteria. And sometimes the metabolites will co-localize with the outer epidermal layer of the root, which is where we expect to see the microbes. So this is just sort of a demonstration of how we're using that chemical imaging to understand the plant microbe interactions. Okay, so some conclusions from this work. What do we know now? Endophytes are fixing nitrogen inside the roots. Not all endophytes are fixing nitrogen equally. Some are having more of a support role to the other cells. But the spatial proximity of these WPB cells to each other matters. There could be some quorum sensing involved. And we're working now to understand what signals the plant might be providing to their microbiome. So with that, I'd like to thank my collaborators and colleagues. And I also want to point out that the rhizochip is a part of EMSL's Terraform's capability, and we're holding a workshop on Thursday. So if you're interested in that, you can still sign up. We have some open spots. So thank you.
Time for questions. Thanks, Jay.
Thank you. Really enjoyed the talk. Could you go back? There was a slide in the late 60s or early 70s in the page numbers. It's where you showed that you thought some of the cells might have been supplying the carbon and others fixing the nitrogen.
I'll try to find it.
That one.
I think there's a bit of a lag with this remote. Yeah, probably. Okay, here we go.
So it's gone.
Sorry.
So is it interesting to me that what you said we're supplying the carbon are all on the inside, and it seems like all the ones that are fixing the nitrogen are on the outside?
Yeah.
There is diffusion going on that plate, right?
Yep.
So do you think that there's anything there related to the way they're doing the same thing within the plant? Is there some fundamental mechanism or spatial organization that's showing you there?
It's a good question. I mean, we were kind of like, why, like exactly what you said, why are only the outside of the plate having this fluorescence? Maybe it has something to do with oxygen. This is an aerobic species. Like maybe there could be better oxygen diffusion on the outside of the plate. We're not really sure yet. We're trying to dig into that and understand like what is regulating exactly the NFH gene. And like you said, there is diffusion going on probably. So the dynamics are going to matter, but what we're thinking is if these cells are like continually producing the polyamines, then they're going to be higher in concentration before it diffuses. So yeah, but yeah, I get what you're saying. The kinetics would matter.
Thank you.
Jayde, very good talk. Following up with the last question, I'm wondering, have you thought about collecting things in the agar, not at the cells, to try to wash or extract to find out what carbon source might be there in abundance in between the cells?
Yeah, that's a really good question. Because I assume that whatever is there is going to be exuded. Yeah. or secreted by the cells. So yeah, that's something that we could probably do to follow on, is looking at exometabolome versus intracellular. Thanks.
The structures you mentioned, nitrosomes, I think is what you called them, are very interesting. What plants do with pathogens sometimes is create these kalos. And so if you look for kalos, which I think is what beta-1,2 glucan, which around these structures so that they could be somehow creating kalos as a way to sequester the bacteria.
Oh, okay, interesting. I didn't know about that. Yeah, we should probably talk after this and maybe get some ideas for what chemicals we could look for. Thank you.